talking about DNA scaffolds or DNA origami, and looking at their interaction with golden nanoparticles. I'll spend the first half of the talk basically going over DNA scaffolds, what they are, how they work, and then also spending time in the second half of the talk going over golden nanoparticles and their electronic interactions with DNA origami and the possibilities there. So first of all, why do we care? Why do we care about DNA? Why do we care about origami in DNA? The first thing, we can actually make really cool synthetic lipid membrane channels biologically. Whether that being like different cells that we've used in the past or examined, or just doing biological modifications on this sort of scale, which is really cool. You can also do different energy transfer photonics, basically looking at either reactions that occur based on photon interactions, or later looking at plasmonic interactions with the DNA origami. We're also looking at molecular walkers and robot facilitation, so making an actual little robot walker of DNA, and then actually using DNA origami as the little road essentially it takes and it moves along in order to, and sort of controlling it in that way, depending on what kind of route you want it to take. So more specifically, going into DNA itself, in case you haven't taken biochemistry or haven't taken a biology class since freshman year, the key fundamentals through Watson-Crick base pairing, having the hydrogen bonds in the DNA, and actually looking at that's a very useful structure. And then looking at being able to modify base pairs through the different using different nucleotides, and actually making like different combinations of the base pairs in order to get unique strands of DNA. And then of course the fact that it's actually a structure that it's a polymer, inherently it's a biological polymer, it's able to actually be used as a building block in a large scale building essentially. And so we have this little smiley face here. Some scientists decided, well, hey, we're going to make some DNA origami. Let's make a smiley face. Why not? So the first thing we have to conceptualize basically an idea target structure, and then go essentially looking at, so DNA helixes are a cylinder type structure. So we can actually use DNA in order to mimic a cylinder display, or in the way that you would make a structure. You use a bunch of cylinders. You can replace it with DNA computationally. You figure it out. And the way you do that is through basically a concept called crossing over, where you have different rows of DNA, and at certain points, we actually cross over DNA in order to form a stable connection, in order to actually have a stable area where you can actually create these structures without them immediately falling apart once they're in solution. Specifically, how this is done is through a viral DNA, a very, very long strand, maybe 7,000, 8,000 nucleotides long, and actually using that as a framework where it's actually the long, for example, in this particular case, it would be the long purple DNA, which is found throughout the entire sequence. And so you use little, what are called frame markers or something like that, and basically combining, and you can purchase these, are maybe like 4 to 5 nucleotides long, or however long you need them to be, 40 to 30, in order to, you can pre-purchase them synthetically, and actually combine them together in order to form a DNA smiley face, which is kind of cool. So going more to the specifics of two-dimensional DNA scaffolding, we have these DNA blocks that we consider, otherwise 10.67 bases, or what we call one turn. Turning being once you reach a certain point, you can actually cross over and connect with neighboring DNA helices. We can combine these together in order to make 16 bases, or essentially 1.5 turns, as your like standard unit, where you actually start having these little blue markers, or these crossing over events that I talked about a little bit earlier along a helix. And so you take an example of structure, a two-dimensional structure. You have these crossover points that occur at various points, and you're actually able to form a stable rigid structure. And having these interlinking points where you have different, again, the fragments of DNA where you want to combine at certain points in order to ensure that stability. And the way this is actually done is you essentially combine the virus DNA chain, the very long black line, of course, and you get your synthetic frame pieces that you put in it. Put it in a test tube, you heat it up to a certain temperature, shake it up overnight. It will essentially combine itself over time. Another way, so we are at two-dimensional. We can actually upgrade to three-dimensional. Picture like a construction worker or something, and you have these various pipes laid out, and you can actually use the same general principle with DNA. Basically stacking DNA helices on top of each other in order to form a three-dimensional structure. And if we take a look at this structure and kind of sort of take several cross-sections along the way, we can see that now instead of just linking or crossing over just in a two-dimensional matrix, you now have a third dimension in order which you can cross over with DNA at several different cross-sections in order to maintain that rigidity and actually create those three-dimensional structures. And if you draw this out and kind of pull it out, zoom it out, you can actually see how you have all these sorts of different linkages. These are essentially a 2D cross-section of the entirety of a three-dimensional image where you have these different crossing over events happening and making sure that the whatever you're trying to design is actually very rigid and very able to stay together rather than just falling apart once you interact with it. So one way we actually do this is through something called the dataless program, and it's actually a way to computationally determine what sort of structure you want, and the program basically automatically designs all the linkers you need for it. So say you have a little shape you want, you're able to fully automatically design the sequence. The program actually determines how it wants to do it the most efficiently possible, and their end result is by creating a DNA-based nanoparticle. And again, like I mentioned earlier, you put your virus scaffold, essentially, your long chain virus, and you have your staples together, and you're able to combine it together, have it fold over time. It will actually stable and naturally, just over time, form into those DNA structures that you want in a sort of auto-assembled or self-assembled process. So one thing that's kind of cool, we can talk about it so we can actually visualize it more or less in real time. We have just different, the white strand is the very long virus chain, you have this linkage, and then over time they kind of create a little shape, and they kind of eventually over time they kind of come together and they form this solid shape more or less, which is pretty convenient and pretty useful in terms of creating three-dimensional structures or structures in general. One method to actually confirm whether or not this method actually works is through cryo-electron microscopy. Ironically, this is actually a method used, or it came up with Nobel Prize last year in chemistry. And so you can actually compare cryo-EM as a method in which you very instantly freeze protein samples with ethane very quickly to the point where they don't crystallize, and you can actually perform electron microscopy with the cold ethane cryo in order to actually image the proteins with several two-dimensional images to eventually forming a three-dimensional image. And if you compare the simulated dataless program or another separate program with the cryo-EM derived, it's actually very similar, and so we can actually very effectively create these structures through computational methods as well, which is really cool. So I mentioned gold nanoparticles earlier in the talk, and we're going to be talking a little bit about that now. So say you have a DNA origami structure and you have a gold nanoparticle. Well, we can actually attach some DNA strands onto the gold nanoparticle, as well as on the inside of a little hole or window within our DNA origami, and in order to attach it. And the connectors between the gold nanoparticle and the DNA strands are little thiol disulfide groups which actually bind to the gold nanoparticle. And so through this way we can actually, and if you see these different colors on the DNA origami, we can actually customize the staple strands on each side in order to have different, like a sort of different puzzle piece, more or less. So depending on how you orient them, depending on what sort of DNA nucleotides or base pairs you use in order to connect, in order on each various side, you can make clusters or cross sections or basically different types of shapes. And so if we actually look at this in a AFM image, atomic microscopy, we can actually see that these show up quite nicely and quite prettily. How generally you just throw these in with the blueprints or with each individual part, and they sort of just auto-assemble or self-assemble over time without really any real issues. And so you can actually see like different particles per cluster and actually compare this to experimentally to simulation in theory in order to see that it actually comes out quite appropriately to both simulation and theory and turns out quite nicely. One thing that people, scientists were like, okay, we have these blueprints. We have Halverson and coworkers were actually able to look at this and they're like, okay, well, let's try and do something more advanced, right? And so they came up with like, okay, we're going to try and make this. But they didn't want to be too complicated, so they made something like this instead in order to kind of simplify things down. But it turns out if you just throw these into the solution with the stoichiometric ratios, then you actually come out with an actual little guy inside your solution, which is really cool. And you're able to, so this just shows the power behind gold nanoparticle design architecture, just as it is. And so we're going to go one step further. So far we've used DNA origami in order to, or we've used gold nanoparticles in order to modify DNA origami. Now we're going to use DNA origami in order to modify gold nanoparticles. So we take gold nanoparticles as a substrate, and we actually design a three-dimensional, what we call a DNA scaffold template. And so this template is actually able to, you can assign different nucleotide lengths of different sizes and shapes and whatnot, and actually bind it to the gold nanoparticle in very specific sites. So before we just kind of threw thiol DNA strands onto the gold nanoparticle at random, but now we can actually selectively choose where we want our bonded strands, as well as figure out what kind of groups we want to attach to it, based on the actual scaffold geometry itself. And so we can kind of expand this outwards. We can attach other gold nanoparticles onto differing lengths and sizes of DNA strands as we want. We can actually attach different things like fluorophore groups, where they can actually be excited and detached at various points in time. So we're essentially using DNA scaffolding on gold nanoparticles, which then can be later used again for DNA scaffolding. So we can get pretty detailed in terms of what we actually want to do. I mentioned earlier, again, we're looking at electronics in terms of DNA. We're going to talk a little bit about hydrogen nanophotonic device application in terms of something called epifluorescence, which is basically fluorescence, but shooting from down above and coming back up. And then precisely looking at cavity intensity. Also looking at jigsaw pieces, which is another type of construction that we can look at and how it affects plasma properties. So nanocavity fluorescence emission, these researchers, Kuszynski and coworkers, decided to create a triangular DNA origami shape. In the center, we actually have, I'm trying to remember the name, cyanine fluorescent markers or something like that in the center, which are actually imaged whenever either they're AFM or for a wide range fluorescence imaging. You can actually get fluorescence out of it and sort of get a intensity out of it. And we have the crystal cavities, where we can actually fit a certain number of these particles inside the cavity, up to 7, basically generating different levels of intensity that we can use, sort of like levels of intensity on a computer monitor. So you have a bunch of different pixels, which the cavity itself would represent, and you can put up to 7 different of these fluorescing DNA origami pieces inside to give 7 different levels of intensity. And so when you put it all together, and you design let's say you have a sheet that you want to manually design, you can actually generate different kinds of images from those intensities and make actual images on the nanoscale level. So this is Van Gogh's Starry Nights, imaged using these fluorescent DNA origami constructions. And we were able to see that in terms of the intensity they wanted to intentionally modify, they were able to do so with quite a degree of accuracy. And based on the number of sites they wanted, and based on, I think it was 65,000 different individual cavities in which they were able to place their desired amounts of intensity in terms of DNA origami. And so another thing, looking at gold nanoparticle interaction with jigsaw puzzle pieces. So again, using our virus, having stable strands, fracture strands, and making another origami structure. Instead, they wanted to see, okay, how can gold nanoparticles, I guess, influence how can they influence the shapes we want to create? So we have different positions here that they indicated as P through 6, P1 through P6. Excuse me. And so taking DNA nanoparticles that were sort of, again, randomly stranded with DNA on these positions in different ratios, we were able to actually form use the gold nanoparticles essentially as a lever or as a fulcrum point where they can connect to the same tips of the different triangular DNA origamis. And this, in terms of plasma properties, we can actually look at C1 is C1, C2, C3 in terms of naming is the different types of gold nanoparticles they use. P is the position they use, and then 20 here is the size of the nanoparticle they use, or the distance away from each DNA origami piece that they use. So using these different shapes that they actually had, they were able to, through AFM imaging, they were able to look and see, okay, we made these structures, and then they looked and they were able to check surface plasma resonance absorption for these various geometries, and saw that depending on the spacing of the gold nanoparticles between the DNA origami, and depending on how they're oriented, you can actually generate specific plasma resonances whenever you fire electrons at the particular binding site. So this could be, in terms of future application, is very promising in terms of varying electronic effects or being able to manipulate it through the DNA origami modulated gold nanoparticles themselves. So in conclusion, there are a really wide variety of ways in which we can modify DNA, and it all kind of started at the beginning of just wanting to make a little smiley face by Rothman. So, yeah, thank you for your time. That's all I have. Any questions?
So they actually just use the computer to design which DNA that will